Earlier, it was thought that carbon dioxide is fixed in plants only by the C3 cycle. But later on, it was found that C3 cycle is not the only cycle operating in all the plants. In some plants, which are growing in tropical regions, plants growing in tropical regions where temperature is high, oxygen concentration is more, and carbon dioxide concentration is less, their C3 cycle is not possible because these conditions are suitable for a wasteful process called photorespiration, which we will discuss later on. In these regions, where in the tropical regions where temperature is high, carbon dioxide concentration is less and oxygen concentration is more, and the plants in this region, they show a special anatomy. The leaves of the plants in the tropical region, they show a special anatomy called kranz anatomy. And these plants with kranz anatomy, they can fix carbon dioxide by using another cycle that is called C4 cycle. Why it is called C4 cycle? Because the first stable compound formed in the dark reactions in this cycle is a 4 carbon containing compound, and that 4 carbon containing compound is oxaloacetic acid. The C4 cycle was given by two scientists, that is Hatch and Slack, in 1967, and this cycle is also known as dicarboxylic acid pathway. तो हमने क्या कहा कि सभी प्लांट्स में C3 साइकिल, पहले हम ये सोचते थे कि C3 साइकिल से ही कार्बोहाइड्रेट फॉर्मेशन होती है, लेकिन बाद में ये देखा गया कि ट्रॉपिकल रीजन्स में जहाँ पे टेम्परेचर बहुत ज़्यादा था, ऑक्सीजन कंसंट्रेशन वाज मोर एंड कार्बन डाइऑक्साइड कंसंट्रेशन इज लेस, their C3 cycle is not possible, and the plants there, they have a special anatomy called kranz anatomy, and in them a special cycle operates, that is the C4 cycle, which was given by Hatch and Slack. Let us see what is the kranz anatomy. The kranz anatomy is seen in both monocots and dicots. Some of the examples of plants with kranz anatomy are maize, sugarcane, amaranthus, etc. Let us see the plants showing kranz anatomy. They have leaf का structure हमें याद है, VS of leaf. This is the upper epidermis. The lower surface of the leaf is bounded by lower epidermis. This is the lower epidermis, and there are present stomata in between, and the space or the space bounded by the two epidermis, इस पूरे region को हम क्या बोलते हैं? This is the mesophyll layer. The plants showing kranz anatomy, they do not have palisade tissue in the mesophyll leaves. पहले हमने क्या देखा था कि mesophyll layer जो थी वो दो parts में divided थी. First there was a layer of compactly arranged palisade cells and then loosely arranged palisade cells. लेकिन plants with kranz anatomy, in them the palisade tissue in the mesophyll is absent, and the cells of the mesophyll tissue, as we can see, they are tightly packed and they contain a large number of chlorophyll molecules. These are the mesophyll cells, tightly packed, without palisade cells, and they have a large number of chloroplasts. The xylem and phloem, जो कि vascular bundle है, the xylem and phloem which forms the vascular bundle, this xylem and phloem, it is surrounded by a sheath of guard cells. जो स्पेशल फीचर है क्रैंज एनाटॉमी का वो क्या है कि वेस्कुलर बंडल जो है दैट इज सराउंडेड बाय layer of special cells, and these cells they are called the bundle sheath cells, and they contain large number of chloroplasts. And the chloroplast in these cells, they are larger in size and they contain starch but they lack grana. इस बंडल शीट सेल्स के जो क्लोरोप्लास्ट हैं वो लार्ज हैं साइज में, लेकिन उनमें ग्रेना नहीं होता. They contain starch grains but they lack grana. Whereas the mesophyll cells, they contain normal type of chloroplast. इनमें जो क्लोरोप्लास्ट है वो नॉर्मल है. और इस C4 साइकिल में the mesophyll cells, these cells, they will perform the C4 cycle, and the cells of the bundle sheath cells, ये वाले जो cells हैं, they will perform the C3 cycle. दोनों ही cycle इनमें operate होंगे, but here the mesophyll cells they perform C4 cycle, and the bundle sheath cells, ये वाले जो हैं, they will perform the C3 cycle. These are the cells of bundle sheath with special chloroplast. So this is the cross section of leaf showing kranz anatomy.